Hello everyone. Today we are going to discuss a new topic, categories of genetic diseases. Now, if you consider any disease, for example, any disease, this disease may have so many pathogenesis, and you can generally classify them as because of environmental characters, or it can be because of genetic, and the effect of each of these may vary. Okay, it can be either small effect or it can be large effect. Now if the genetic component has a larger effect, then we call them generally as genetic diseases. Now genetic diseases, we can divide them into three types. So the first one would be chromosomal disorders. Now what happens in chromosomal disorders is there is a change in number of either the entire chromosome or segments of chromosome. So either the entire chromosome can be duplicated or deleted. Okay, so it can be duplicated, replicated as well, or it can be deleted. An example which I can give is trisomy 21. So we already know this to be as Down syndrome. Here what happened to individual genes, they are actually normal, they are not abnormal. Rather, the entire chromosome either appears in more numbers or it is not appearing at all. So this is the problem. Now in if the same thing like deletion or addition or duplication happens in smaller segments of chromosome, we call that as copy number variation. That idea is what we call as copy number variation. So one example of this kind of disease is DiGeorge syndrome. So here what happens, it is generally written as 22q11.2 deletion. So what happens, a small segment of genes present in 22nd chromosome in the long arm or Q arm at 11.2 position is being deleted. So only a small segment is being deleted. So because of this, you have multiple genes are affected or not present. Because of that, you have a syndrome, and that syndrome is called DiGeorge syndrome. And if you see, chromosomal disorders are the reason for about 50% spontaneous abortions. So that is about the chromosomal disorders. Next up, we have something called as single gene disorders. So unlike an entire chromosome or segments of chromosome, a single gene is being affected. Only one gene is affected here. This is also called as monogenic disorders, or otherwise also known as Mendelian disorders. And depending on how many allele is affected, it can be two types. It can be homozygous when both the alleles are affected, or it can be heterozygous when only one genus or one allele is affected. Now the characteristic features of this is that they show what is called as classic inheritance pattern. What does it mean? You can classify these disorders according to the pedigree analysis. So they would either reflect as autosomal recessive, autosomal dominant, or X-linked disorders. Okay, so you can easily categorize them into one of these. And mitochondrial DNA can also be affected. So this includes mitochondrial diseases as well. So not just your nuclear DNA, but mitochondrial DNA can also be affected, and it is also included in single gene disorders. So first we saw chromosomal disorders where an entire chromosome is affected or only segment of chromosome is affected. If only segment is affected, we call them copy number. Here we have a single gene disorders. They can be either homozygous when both alleles are affected, or only one allele is affected. We call them heterozygous. They show classical inheritance patterns, either recessive, dominant, or X-linked, and mitochondrial diseases also included. Then nowadays we have a third set of disorders which are now becoming more and more prevalent, and they are called as multifactorial disorders. They are multifactorial diseases. Multifactorial, that means we have already discussed here about diseases having environmental factors as their cause of pathology or genetic causes. Now in multifactorial, both will have significant involvement, so that is why they are considered as multifactorial. So now what happens is that they will have complex inheritance. That means you know for sure they are genetic diseases. Genetic involvement is definitely there, but you will not be able to point to which gene or chromosomes. This is because unlike the earlier two, where either a particular chromosome is affected or a particular gene is affected, here multiple genes in multiple chromosomes are involved. So if you consider a chromosomal disorder, there you will have a single chromosome, and either the next chromosome will be deleted or the same chromosome would be duplicated multiples of times, right? Or what happens, a particular segment will be duplicated multiple times or deleted. Now in single gene disorder, what happens, you have a chromosome and one particular gene, one single gene is being affected. But here what happens, you have one chromosome and another chromosome and a third set of chromosome, multiple genes from each of these chromosomes would be affected. That is why it is very difficult to diagnose and treat these conditions most of the time, and there will not be any proper inheritance pattern. No proper inheritance pattern. So you will not have any classical inheritance pattern like this. Instead, you know that the incidence is very high in close relatives of the deceased people, or you know that the chance of a close relative getting the disease is very high, But other than that, you will not know who will get, when they will get, how they will get. Those things will not be there. It will skip at irregular intervals, and it will appear back again at in irregular intervals. Pedigree analysis will not yield a very good idea about the type of the disease. That is also because of this characteristics only. This is this, and also because of involvement of environmental factors. So there is significant environmental involvement, so that could also be there. So some examples of this, one of the best example which I can give is diabetes. So everybody knows. Then we have inflammatory bowel diseases. Then we have various types of cancers, right? Breast cancer, ovarian cancer and all that. And then congenital malformations can also happen, like congenital heart diseases or cleft lip. So these are all some of the examples of multifactorial diseases. So genetic diseases can be simply classified as chromosomal single gene multifactorial diseases.